Hello students, welcome back. We're going to continue with the nervous system part 4. And remember, we're talking about just one aspect of the nervous system, the sympathetic component of the peripheral nervous system. The sympathetic component of the autonomic component of the peripheral nervous system. So let us continue where we left off. This is nervous system part 4. Now last time we were trying to figure out what type of receptors. Remember, there are two types. Let's go back here to our review. There are two types of adrenergic receptors. Remember, the typical adrenergic compound is epinephrine. That's going to be our parent compound all the time, epinephrine. And we have two types of adrenergic receptors, basically. We have sympathetic adrenergic receptors, which are found on the organs or the tissue that needs to be stimulated, and we have inhibitory adrenergic receptors, which are found on the tissues that need to be inhibited. And the rule is, I gave you a rule. For the stimulatory adrenergic receptors, they're going to always be alpha 1, except for these three situations: the heart, the juxtaglomerular apparatus, and adipocytes, fat cells. They're going to be beta 1, or a type of beta 1. And for the inhibitory receptors, for the tissues to be inhibited, the receptors are going to always be beta 2, with the exception of with three exceptions: presynaptic nerve endings, platelets, and the pancreas. They're going to be alpha 2. So now we're going to go through our five tissues that we identified to receive a sympathetic response, and try to predict what type of adrenergic receptor is found there. Okay, so let's go back there. So we went here. I think the last one was here. Okay, our pilorector muscle. Our pilorector muscle has to contract to stand the hair up. So, do you need to have an inhibitory receptor here or a stimulatory receptor? Stimulatory receptor. So there are two types of stimulatory receptors: alpha 1, beta 1. Which one is it going to be? Alpha 1. Very good, because it's not one of the exceptions. Excellent, excellent. Okay. Let's do the next one here. For your pupillary dilation. Okay, you want your pupil here. You're going to want your pupil to. Do you need for your pupil to? In order for your pupil to get larger, do you need for your pupil to, the muscles in the pupil to contract or to dilate? You need them to contract or relax. Which one do you need? You need them to relax. Okay, and so are you going to want? So if you want relax, do you need inhibitory receptors or stimulatory receptors for relaxing? If you want something to relax, it needs to be an inhibitory receptor, because you don't want it to contract. So there are two types of inhibitory receptors: alpha 2, beta 2. Which one is going to be found here? Beta 2. Now, your rationale is right. You're thinking correctly, but we're going to come back to this. We have to do something a little special for this one. We'll come back to it. Okay, but we'll just get the understanding. So I'm going to go ahead and put here. I'm going to put beta 2 here. Okay, you all said beta 2. I'm going to put a question mark. Is that right? But we'll see. We'll come back to that one. Now, for the thinning of the lens, okay. Now, there are muscles that actually pull on that lens there. That ciliaris muscle holds the lens in place using these suspensory ligaments. Now, do you want these muscles to thin the lens? Do you want these muscles to contract or relax? You want them to contract. And so do you want inhibitory receptors or stimulatory receptors? Stimulatory receptors. And so there are two types of stimulatory receptors: alpha 1 and beta 1. Which one is it going to be? Alpha 1. Your rationale makes sense. Okay, and let's put here, I'm going to put alpha 1 here. We're going to come back. We're going to come back to this one though. Alpha 1 question mark. Because the eye is a things in the eye almost. Eye tries to confuse students. Okay, let's look at these here. So your vascular constriction and relaxation. Now, in the you want to be able to give blood basically to your stomach. And I'm going to actually draw a blood vessel here. So it's going to be a blood vessel coming from the back and coming up to the front. Okay, here's our blood vessel here. And within the blood vessel, you're going to have some smooth muscle. Okay, now if you want to close off those blood vessels that are going to the stomach, you want to close those blood vessels. Do you need for those smooth muscles in these blood vessels do you need them for them to constrict or relax? You need for them to constrict. And so do you need inhibitory receptors or stimulatory receptors? Stimulatory receptors, that is right. There are two types of stimulatory, alpha 1, beta 1. Which one is it going to be? Alpha 1, exactly. Does that make sense? Now, there's nothing else to say about this one. This one just makes sense. So this is alpha 1. You can see the same. It's going to be the same case for the kidneys because they have to constrict. So what type of receptors are going to be here? Alpha 1. For your skin, same situation. What type of receptors? Alpha 1. And over here, now for your muscles, your smooth muscle, there are blood vessels which supply blood to your skeletal muscle. Do you want those? You want those blood vessels do you want those blood vessels to dilate or to constrict the blood vessels? You want them to dilate so you can get more blood to your skeletal muscle. Okay. The smooth muscle. So we're talking about two different muscles. This one can be a little confusing. So I'm going to draw here. Okay. Okay, and then you're going to have your smooth muscles. Your smooth muscles are in the blood vessels which supply blood to your skeletal muscles. But we're talking about the smooth muscle now. Now, do you need for this blood vessel, do you need it to dilate or constrict the blood vessel? If you want to give more blood to your skeletal muscle, do you need for that blood vessel to get larger or smaller? You need to get larger, exactly. Now, the smooth muscles in there. Do you need for those smooth muscles in that blood vessels, do you need that smooth muscle to contract or relax? You need it to relax because when that smooth muscle relaxes, it's going to let the blood vessels stay open. Does that make sense? Okay. Now, so if you want the smooth muscle to relax, do you need to have an inhibitory receptor or a stimulatory receptor? Inhibitory receptor, because you want to inhibit that smooth muscle. You don't want it to contract. Does that make sense? That's a key point. So there are two types of inhibitory receptors. There's alpha 2 and there's beta 2. Which one is going to be here? Beta 2, because beta 2 is the default. Makes sense? Very good, very good. You all are on top of things, on top of things. And we don't have to put a question mark because that is the case. Let's see here. Okay, let's do this one here. Okay. In our bronchioles, okay, what I'm going to do here, I'm going to draw a bronchiole here. Let's see here. I'm going to just draw a bronchiole. This is a bronchiole here. Okay. And within the bronchiole, you have your smooth muscles. Now, the question is, when you're under sympathetic control, do you need for your bronchioles, if a dog is chasing you, do you need your bronchioles to get larger or to get smaller? You need to get larger. Why do you need them to get larger? So you can let more air in. Very good. Now within those bronchioles, there are smooth muscles. Do you want those smooth muscles to constrict or relax? You need for them to relax. Exactly. And so if these smooth muscles are going to relax, do you need an inhibitory receptor or a stimulatory receptor? An inhibitory, because you want to inhibit their contraction. There are two types of inhibitory receptors. There's alpha 2 and beta 2. Which one is going to be found here? Beta 2. Very good. Make sense? Excellent. And we only have one more to go now. I have to make sure this makes sense to you. Okay, here we are. Cardiostimulation. That was the other one. Now, when you're under sympathetic control, a dog is chasing after you. Do you need for your heart to beat faster? Do you need more cardiac output or less cardiac output? You need more cardiac output. So do you want to stimulate the heart or inhibit the heart? You want to stimulate. So do you need a stimulatory receptor or an inhibitory receptor? A stimulatory receptor. There are two types of stimulatory receptors: alpha 1 and beta 1. Which one is it going to be? I didn't get you. You all remembered the exception. Exactly. And so the heart is going to be beta 1. And if you understand that concept, that there are different receptors on different tissues, you understand the sympathetic nervous system. Make sense so far? So now let's go back and look at those special cases. Not really special cases, but just really need to look at the eye. The eye is the one that gives us the most problems. Okay. So I'm just going to go up here on a new slide and we're going to do the eye. So we'll put this in today's lecture. Okay. Okay. So here we're going to have ourselves we have ourselves an eye. And within the eye, we have our pupil. Now, the special thing about the eye is that the eye, well, let's just do it this way. What do we say our answer was for the eye? I'm going to go back to this first, and so it'll make sense. What type of receptor do we say was going to be in the eye? So beta 2, very good. So this is the way you get, okay, in the eye or in the pupil, you have these smooth muscles in the pupil. And these muscles are called constrictor pupillae. So this is I'm going to point to it with this color. This muscle is a constrictor. This is the name of the muscle. Pupillae. Okay. And so when this muscle is activated, it squeezes the pupil and makes it smaller. Does that make sense? Now, when you're under sympathetic control, do you want your pupil to get larger or to get smaller? A dog is chasing after you. Do you need a bigger pupil or a smaller one? You need a bigger pupil. And so for this muscle right here, do you need for this muscle to contract or relax? You want it to relax so that the pupil can get larger. And so do you need an inhibitory receptor or a stimulatory receptor here? Inhibitory receptor. And so there are two types of inhibitory receptors: alpha 2, beta 2. Which one is it going to be? Beta 2. Does that make sense? Now for the twist for the eye. This is the twist. The eye also has another type of muscle. It has radial muscles. So it has smooth muscles that go this way. And these muscles are called dilator pupillae. These are called dilator pupillae muscles. And so when these muscles contract, they get shorter and they pull the iris, they pull the pupil open. They pull the pupil open. And so when you're under sympathetic control, do you want these muscles, the dilator pupillae, do you want them to contract or relax? You want them to contract because when they get smaller, they're going to open up the pupil. Make sense? So if you want these muscles to contract, do you want to have inhibitory receptors? Well, do you want to inhibit these muscles or stimulate these muscles, the dilator pupillae? You want to stimulate them. So do you want stimulatory receptors or inhibitory? Stimulatory. There are two types of stimulatory, alpha 1, beta 1. Which one is it going to be? Alpha 1. Very good. Very good.
So you need an alpha and a beta to make the eye dilate?
Exactly. In this situation, if you give if epinephrine goes to a person's eye, you drop epinephrine on a person's eye, the epinephrine is going to go to the alpha 1 receptors on the dilator pupillae to cause it to relax, cause them to contract and open the pupil, and that epinephrine is going to go to the constrictor pupillae to inhibit them to cause the pupils to dilate. So you only need one drug like epinephrine would do both of those things. Any sympathomimetic drug is going to cause the pupils to dilate. Make sense? And so you have to remember, epinephrine does both things. It inhibits some receptors and it stimulates others, but it does it to the right ones. Excellent. And I think now that you have an understanding of how the receptors. I was going to talk about how the suspensory ligaments and the lens works, but I think you understand the situation pretty well now. Now let us look at the biochemistry of this situation. What happens biochemically to get the signal from the outside, from the tissue, to the inside of the cells to give you a response? That's what we're going to look at now. Okay, let's see which one of these. We have 5 organs we could look at. Question?
Yeah, you questioned the thinning of the lens.
Yeah, I did, but I didn't want to. Yeah, let's go over it. Let's go over it then. Because that's a very special situation, that thinning of the lens. And, well, I guess you should understand. Let's do the thinning of the lens. I wasn't going to do it, but because the thinning of the lens is a very special case. It doesn't make as much sense as the others. Okay. Let's do it like this. Okay. You want to watch me draw this first because it takes me a long time. I have to look at my old notes to see how I drew this. Okay. Oh, that's not right. Go this way. Okay. Okay. There's the eye. I'm going to put here. This is the cornea of the eye here. And I'm going to put here. This is going to be the lens right here. This is the lens of the eye. I'm not going to make it so big because I need some space. Okay, this is the lens. Remember, what holds the lens in place is closer to the front, but I just need it to be here for now. What holds it to the front are going to be these things called suspensory ligaments. Here's a suspensory ligament. Okay, those are your suspensory ligaments. Do I need to label these parts, or are you all okay with? Okay, let's label them then. Okay. Okay. Here, this is the cornea. Yeah, because everyone may not have taken A&P. That's the cornea. I need another color. This here is a suspensory ligament. Suspensory ligament. What is this here? That's the lens. Now, the suspensory ligaments are attached to a muscle. Okay, I'm going to make this muscle red. Anybody remember what the name of that muscle is? That's okay if you don't remember. Okay, let's put this muscle here. This muscle is the ciliaris muscle. I'm going to label it in just a second. Okay. Let's get a good color here. There's a concept here, and I'm hoping that you'll get the concept. I tell you. How can I do this? Anybody have a rubber band? Rubber band? Anybody? I know nobody has one. It would only be great if someone did. Oh, may I borrow one? May I borrow? Pass me, please. Great. Oh, you have a good long one. Excellent. Excellent. Okay. Now, I need you all to think about it like this. Okay. Okay. This is the lens. When the lens is thin, it's like this. It's stretched out. Now, one way you think about it, you can think about it like this. If this is the lens, and then when the ciliaris muscle contracts, the lens gets thinner. Make sense? But that's not what happens, because it's not designed like that. Think about it like this. Okay, this is the lens here under... Let's see here. This is the lens when it's thin. Now at this point, when the lens is very thin, the ciliaris muscle is relaxed. It's relaxed. I know it's hard to think about it like this, but now when the ciliaris muscle contracts, it pulls forward, and you can see it on the diagram. It pulls forward and down. And up this way. And when it contracts, it pulls the lens in. It pulls in like that, and it makes the lens get thicker. So when the ciliaris muscle is relaxed, the lens is thick. But when it contracts, it pulls in, the lens gets thicker. Okay, let's look at it on here. Now, this is the ciliaris muscle here. When the ciliaris muscle is long and relaxed, the lens is going to be thin. But it's almost like the lens is like rubbery. How can I do this? How can I do this? Where's that other one? Okay. It's like, okay, this is the ciliaris muscle. And so when it's, I don't know if I can see myself. Okay, I won't put it so hard. Okay, this is the ciliaris muscle. And so when the ciliaris muscle is relaxed, the lens is thin. But then the ciliaris muscle contracts, it comes in like this, and then the lens gets thick. When the ciliaris muscle contracts, that thickens the lens. And that is not intuitive. That's so opposite of the way you would ever think of any muscle doing anything. And so you just have to remember that this is just a very stupid exception. So when the ciliaris muscle is relaxed, the lens is thick. But when it contracts, it pulls in, it pulls front to the front and in, and then the lens just thickens up. It blows up outward. Does that kind of make sense? It goes against reason. So you have to just remember that exception. Is everyone okay with that? Okay, now. So, now let's look at and see what's going to happen here. So, under sympathetic control, do you want the lens to be thick or thin? You want it to be thin. Why? So you can see far away. Why? Why do you want to see far away? So you can escape. Very good. So you want the lens to be thin. Now, this is the trick we want here. Do you need for the ciliaris muscle to contract or relax? Exactly. That's the exception. It makes no sense unless you understand this diagram. You need for the ciliaris muscle to relax. So if you want the ciliaris muscle to relax, do you need an inhibitory receptor or a stimulatory receptor? You need an inhibitory receptor. So, do you need for that receptor? There are two types of inhibitory receptors. Alpha 2, beta 2. Which one is it going to be? Beta 2. Beta 2. Make sense? It's the biggest. Wow. Which one did we put for ciliaris? Right, because if you think intuitively, if it contracts, you're going to pull the lens thinner. But that's not how the ciliaris. The ciliaris is a great exception to everything. It follows the same rules, but it's just how it's designed. It's because of how the eye is designed. Does that make sense? So the receptor on ciliaris is going to be beta 2. Okay, everyone okay with that? Now that we know how all the receptors work, we know why you have sympathetic responses. Now we're going to look at the biochemical mechanisms. We're going to start looking at the biochemical mechanisms that lead to these responses. And we've looked at 6 tissues, but I want to look at the heart first. Let's look at the heart first. Okay? So, yes. Now, like on the parasympathetic conditions, like right now, your lens is going to be globular. It's going to be big now. That's we call that normal. We call it parasympathetic normal because if you're just comfortable sitting down, now just relaxing, that's parasympathetic. Only when you're faced with a very stressful situation do all of these sympathetic responses start taking place. Again, that's going to be most of the time, sleep is going to be because it's rest and digest. So sleep is rest. So that's going to be parasympathetic. And if you're having a bad dream and it's caused you a lot of anxiety, a lot of stress, then you're going to have some sympathetic responses taking place even while you're asleep. So it's all dependent upon if epinephrine is produced or not. If the adrenal gland is producing epinephrine, then you're going to get some of those sympathetic responses. Question for you all, just to see if you understand things. The act of sexual arousal, is that going to be a parasympathetic response or a sympathetic response? Which one do you think it's going to be? Parasympathetic sexual arousal. Okay, very good. Sympathetic, you all say? Now, in actuality, it is parasympathetic because you have to be, for a male to be aroused, you have to be relaxed. In order for a female to produce secretions, she has to be relaxed. She can't be stressed out. She's worried about things, or if he's worried about things, you aren't going to get the arousal. Make sense, kind of? So remember, those are going to be sympathetic responses. However, for the ejaculation and the orgasm, those are going to be sympathetic, extremely sympathetic responses. Okay, let us continue. Now let's look at what organ did we say we're going to look at? Which one? The heart. Let's look at the heart. So let us draw for ourselves a cardiac cell. So I'm going to draw here a cardiac cell. I'm going to let it take up the whole. I won't get you all of these. Okay. a cardiac cell. Now, what type of receptor is found on cardiac cells? Stimulatory or inhibitory? What type of adrenergic receptor? Is it going to be stimulatory or inhibitory? Stimulatory. So stimulatory is alpha 1 or beta 1. Which one is it going to be? Beta 1 is the exception. Okay? So let's draw for ourselves a beta 1 adrenergic receptor. Let's see here. What would be a good color? Okay, I'm going to draw my receptor like this. I'm going to give it a little cup shape here. I don't want that. Okay, and it's going to go through the membrane 7 times. 1, 2, 3, 4, 5, 6, 7. Okay. So this is my beta 1 adrenergic receptor. That's the receptor on a heart cell. So biochemistry is really about mechanisms and pathways. But I didn't want to teach you the pathways before you knew why you needed them, because that would be pointless. You wouldn't see a rationale for it. Okay? Now, that's the receptor. The chemical, does anybody know the name that we give to chemicals that bind to receptors? There's no way you'll know that. Okay. The chemical that binds to a receptor is called a ligand. Probably never heard that word before. Is a ligand. And so let's put here our ligand that binds to our, this is our ligand that binds to our beta 1 adrenergic receptor. Now, what is the name? What is the name of the chemical? What is the name of the adrenergic chemical that we're going to use for epinephrine? So this chemical is going to be epinephrine. So here we have epinephrine. Okay, so epinephrine is going to bind to our beta 1 adrenergic receptor. Okay, so I'm going to go back out here. Now, within our heart, you're going to have some fibers here. You're going to have some muscle fibers within our heart cell. I'm going to draw one here. Draw this and this. And it's going to have lots of muscle fibers. Okay. So that green muscle that we have, that green protein here, that's going to be myosin. Bless you. And the blue protein is going to be actin. It's going to be actin. Okay. And what's going to happen here, in order for the heart to contract, those myosin filaments are going to grab the actin and pull them in and cause that muscle cell to get shorter. And when that muscle cell gets shorter, all around the heart, that's going to squeeze on the ventricle and push the blood out of the ventricle. Make sense? And so that's going to be the contraction of the heart. And so there are going to be a lot of steps. Okay, you don't really have to draw. There are going to be a lot of steps here to go from epinephrine binding to the contraction of those proteins to squeeze the heart. And when we come back next time, we're going to figure out what those steps are. Does the rationale make sense where we're going to go with this? And that's called a biochemical pathway. Lots and lots of biochemical pathways we're going to learn about. But I need you to understand the rationale. Epinephrine causes the heart to beat faster, causes more cardiac output. In order to get that cardiac output, you need for your heart, your muscles in your heart, or the muscle proteins in the heart, to actually overlap one another to constrict or shorten those cardiac cells. And that's what we're going to learn about. What happens? What are those steps that lead to the contraction of that heart cell? Make sense? Any questions? Any questions about this biochemical pathway? Question? Okay, we're going to go over that now. So I'm going to stop this here.